Hello everyone, welcome to the Wise Tutor YouTube channel. Today's video is our 5th video in the AP Biology sequence where we're going to be talking about proteins, our 3rd macromolecule. So proteins have a variety of functions within the body, honestly too many to list, but I just listed a couple. They give structural support and protection and defense for a lot of organs and structures themselves. They also help transport substances and nutrients around the body. And an example I put in quotations or in parentheses here is hemoglobin, which is a protein that transports oxygen and carbon dioxide in the bloodstream from tissue to the lungs and back. So that's a pretty important protein in the body. Movement, proteins also facilitate movement in a sense. They also are in charge of regulation in many instances, with an example being hormones. Hormones are actually proteins, so that's important. And catalysis, which we'll talk about more in our enzymes unit, but catalysis is basically speeding up a chemical reaction in the body. And enzymes are very, very important for biological functions. So proteins, they have a monomer of amino acids. There are 20 amino acids, 20 known different types of amino acids in humans. Their polymers are a dipeptide or a polypeptide. Again, you're seeing very similar throughout the unit the prefixes. So di meaning 2 peptides, poly meaning 3 or more, and we mean by the numbers we mean 2 amino acids or 3 amino acids or more for the polypeptide. Now the bond type that goes in between each amino acid is called the peptide bond, and I wanted to show you guys that right here. It's the one in blue. On the next slide, we'll talk more about the structure of an individual amino acid, and you'll see the NCC chains. Becomes actually a very good way of distinguishing and identifying proteins among other macromolecules. So when we talk about the peptide structure, we are talking about the NCC backbone, NCC. We're also talking about an amino group of NH2. So this is called an amino group. It is one of the 3 parts, or 4 parts really, of the peptide structure. You've got, next is the carboxyl group with COOH minus. So you've got the OH minus here. You've got a C and O here. So this entire thing is called the carboxyl group. And then you've got the central carbon here, or what we call a central carbon in the amino acid. And then the last kind of part of the amino acid is a side chain or a functional group. And those are usually denoted on generalized sketches and structures, really. They're denoted with an R. So those are the functional groups. Now remember how I said that there were roughly 20 discovered amino acids for humans? Here they are actually, and really the way that they differ is that they differ in the R group, or the functional group, or the side chain. Each one of these like molecules drawn here represents a different side chain, and you see that each one has some sort of property. For example, these are nonpolar. These 10 are nonpolar. These 5 are polar. You've got 3 that are charged and positively, and 2 that are charged negatively. So this becomes very, very important when we begin talking about protein structures, which we will in a minute. But I just wanted to make sure that you know that the variations of the amino acids really come due to the variations in this R group, because everything else in an amino acid is the same. You've got the NCC in all of them. You've got an amino group and a carboxyl group in all of them. But the only thing that differs is the side chain. We'll talk about that. So when we talk about structure of proteins, we've got 4 structures, okay? It's important to memorize these. That's why they're highlighted in red, or their text is in red, because they're really important to know. So the first structure that we're going to talk about is the primary structure. First, primary. And the primary structure is the sequence or chain of amino acids bonded together by peptide bonds. So here's a picture of that. You see this long chain of these amino acids. Each circle represents a different amino acid here. and the black lines connecting each circle or each amino acid is a peptide bond, which we just showed in the previous slide. And here again, we've kind of taken a couple of these and we made them into their molecular form. Now again, just for identification, you could go ahead and identify what each of these structures is. You've got NCC here, NCC here, NCC here, and so on. And you could actually count how many amino acids this chain is made up of. And so that's a good way to distinguish that. The secondary structure is when hydrogen bonds form one of two structures in a larger protein, usually. And those are an alpha helix or alpha helices for plural, and beta plated sheets. All right. So usually we don't write alpha and beta like this. We actually denote them with their Greek letters, symbols. But here is a really good picture that shows these structures. So you could imagine a protein looking like this. Now we kind of translated that to the exact shape. All right, so this is an alpha helix. You see it's helical in structure, but the important thing that I want to show you is the primary structure in this is retained. You still see the NCC. You still see the NCC going all throughout the alpha helix, but now you've got connections between the spirals, and you've got these connections due to the hydrogen bonds that are present among the amino acids. And so this is important for the alpha helix. Now, again, we've got the second structure here, which is the beta plated sheets. And you could see potentially an application for that right here. This would be an example of a beta plated sheet. And you still see the NCC amino acid sequences here, or chains, but you see that different interaction with the hydrogen bonds that weren't originally present in the primary structure. And so that's really important to know. You need to know these two structures. They're very important. And you also need to know that hydrogen bonds are responsible for creating these structures. The third one is the tertiary structure. And that's when the protein begins to fold and bond together. So the peptide chain is folding and bonding, and it gives the protein its 3D shape. All right, and the way that the 3D shape is usually determined is highly dependent on the properties of the functional groups found on the amino acids of the primary structure. So this will have us refer back to the primary structure. We said in the primary structure, you've got these amino acids and you've got these R groups. Okay, let's go even one slide back. You've got these variable R groups, all right? So what happens is in the tertiary structure, you see different kinds of interactions occurring between the R groups of different amino acids. And that makes the peptide or the protein fold in certain ways. All right, so for example, we've got 4 different examples here. You could see salt bridges between the R groups. You could see hydrophobic interactions between the R groups, meaning that they want to come closer together because they're afraid of water, hydrophobic. You've got disulfide bonds and sulfide sulfur. So those usually occur when the R group contains sulfur. And then finally, you've got hydrogen bonds that could form between them as well. So those are all different examples of some of the interactions that make the protein fold and contribute to its 3D shape. So we don't see really like this kind of thing happening in the primary and secondary structure as well. Now the fourth structure is called the quaternary structure, and that is when that is the bonding of 3D subunits. And what are subunits? How are they defined? Well, subunits are just polypeptides that have a tertiary structure. And so what happens is they bond and form a larger or more complex protein. All right, and it's important to note 2 things. Not all proteins have a quaternary structure. Okay, only big proteins do, and only proteins made of more than one polypeptide do. So they could have up until the tertiary structure, but it's not necessary for a protein to have a quaternary structure. Obviously, for the bigger ones, they must have it. That's how they function. but that's really important to know. The second one is second thing to note is that the quaternary structure and how it folds and curls up, like as shown in this diagram, is highly dependent on similar subunit interactions as the subunits that we as the interactions that we talked about in the tertiary structure. All right, so you can see here a diagram of a quaternary structure of a protein. You've got several different subunits denoted by these different curls and different colors. And each time they curl, you could assume that there is some sort of subunit interaction between the two subunits. For example, when you see like this interaction here, right here between the purple and the violet and like the purple colors or indigo color, you see it, the indigo bend a bit, the subunit bend a bit, and that is due to an interaction between this subunit and this subunit. And you see that structure in a lot of complex proteins. And most of the structures, most of the proteins that are, that we're trying to research about and discover are very complex like this. There are not many proteins that have less complex structures. All right, and I hope you enjoyed the video about proteins. Keep on a lookout for the nucleic acids video, and we're getting really close to finishing this unit, so I hope you enjoy.